Welcome to the start of our histology chapter. Now we're going to spend some time looking at the key types of tissues that we find in the human body. And just like many of our chapters, this is not a complete histology course. Our goal is really to get an appreciation for the form and function of some of the basic tissues like epithelial, the various types of connective tissues, muscles, etc. Now, it does take a lot of practice to really get really good at looking at histology slides, but we can see at least some of the basic tissues. Now, the one thing I want to point out are some really key concepts in that often in the world of histology, they called it like they saw it. And as you can see in this diagram right here, we have a number of different tissues that are actually part of this slide. Because the thing to think about is that when we go to look at a tissue, the tissues really are taken from organs, and organs by their very definition have two or more tissue types. So often when you first look at a slide, especially at low power, you could see 2, 3, or 4 different types of tissues on that slide, and the trick is to figure out what you want to look at in particular. Do I want to be looking at the connective tissue in this tissue, or do I want to be looking at the epithelial tissue? Now, I like this slide because, of course, if you look at it long enough, I can see a dinosaur and you can probably see it too. And it's a good reminder that we call it like we see it in the world of histology. Now, just to start, there are about 50 trillion cells and about 200 different types of cells within you. Now, we can break those into the tissues into some major groups. Epithelial, connective, nervous, and muscular. Each one of these major groups has subtypes within it that we'll have a look at. And each one plays its own unique set of roles, and each also has its own characteristics. And what you want to do is develop sort of a checklist as to what are the key characteristics epithelial will have versus connective versus nervous versus muscular. So when you start to look at the slides, you already have a preconceived notion of what you should be looking for. Now, just want to mention organ is a structure with discrete boundaries composed of 2 or more tissues, and of course histology is a study of tissues and how they're arranged. Now, a tissue is a group of similar cells or cell products working together to perform a specific role within an organ. I think that's a pretty good definition, and we already said there's 4 primary types: epithelial, connective, nervous, and muscular. Now, how do these differ from each other? Well, it differs in terms of the types of cells we find in there. Epithelial cells will have certain groups of cells. Muscular tissue will have different types of cells, and it's also about their products. So it's the type and functions of cells, and also the matrix. Now, what's the matrix? The matrix is, here we have cells within a tissue, and the matrix is the material basically surrounded, outside the cells. It's the material that the cells exist in. Now, in some tissues, it's basically almost all cells with very little visible matrix. In other tissues we're going to see, there's much fewer cells with a lot of matrix. So the ratio of cells to matrix often gives you an indication. And we can see that here the relative amount of space occupied by cells versus matrix varies from one to the next. Now, what is matrix? It depends on the tissue type, but some generalities about the matrix is that we will see fibrous proteins, and we'll also see this clear gel called ground substance. Now, I want to warn you, ground substance goes by a whole bunch of other names. It's known as tissue fluid, extracellular fluid, or interstitial fluid, or tissue gel. So there's a lot of names you can hear it called by. Now, before we get right into the tissues, we should realize everything came from embryonic cells and tissues, or the primordia. So a fertilized egg, of course, becomes an embryo with layers, and we always talk about the three primary germ layers: ectoderm, which was the outer layer; endoderm, the inner; and mesenchyme, which became the middle layer between the two. Now, these give rise, once you're a group of cells that have ended up either in ectoderm, endoderm, or mesoderm, your fate has been partially determined. Because if you're ectoderm, you're going to give rise to the epidermis and the nervous system. The endoderm, so if you're a group of cells in the embryo that end up in the endoderm layer, you give rise to mucous membranes that line the digestive respiratory tracts and the digestive glands, and some other tissues. And if you happen to end up in the mesoderm, then you basically give rise to cartilage, bone, blood. So these tissues tend to have these wispy collagen fibers and a group of cells called fibroblasts, which make the matrix. Now, you have to remember, when we're looking at any type of tissue under a microscope, that tissue has been highly processed. We've taken it out of a three-dimensional structure, and what we've done is we've had to prepare it so it doesn't basically decay. So water is drawn out very slowly through a series of water alcohol baths. Then you have to replace the water with other materials, and then you have to harden the tissue by slowly embedding wax into it over a period of time. And then you also have to stain the tissue so you can actually see it, and different types of chemical stains are used. So by the time you get to look at the tissue, it's undergone quite a bit of processing. So we preserve, slice, and section tissues. We use a fixative like formalin to keep it from decaying. We slice the tissue into very thin sections of just 1 or 2 cells thick. If it was much thicker, the light wouldn't pass through it on the light microscope. And then we stain it, and then which artificially colors it with histological stain. Now, there are a number of stains out there: hematoxin, eosin, right stain, etc. The list goes on and on. We'll be seeing a few different types of stains, but some stains will bind to different cellular components. So really what we've done is we've taken a 3-dimensional object and turned it into a 2-dimensional object and have highly processed it. Now, this processing sometimes, of course, can lead to artifacts. If a knife is dull, often you'll see you can actually see that the tissue looks almost like it's being chopped up. Or often if a stain gets old, there's barely any color into the tissue. So those are things we call artifacts. Now, let's have a look here. I think this is a good diagram. Here's things that like let's take the example of an egg. That if we were to section the egg here, we would, if that's all we saw from an egg, we'd get an impression that was fairly small. There seems to be nothing in it. If we section here and then look, then we can see, all of a sudden it looks more like it's kind of not quite an oblong, but sort of looks sharp at one end, and there's something inside. Section it here, it looks much bigger, etc., etc. So this gives you a reminder that you can rarely just look at just one section from a tissue to really get its overall appearance. That ideally, if you had the time and you really want to understand a tissue inside out, you would actually look at a number of conjoining sections to really develop a 3-dimensional structure. Same thing here with tube structures. If you cut it here, then you'd have no idea it looks like a tube. If you cut it here, you may not be able to tell that it's actually this shape. And if you cut it up here, you may actually think it's 2 different tubes. So it's easy to get a misrepresentation of what a structure looks like. Or over here, we can see here is like a sweat gland going up to the surface. You can see it's one long continuous tube. But if I did a certain section, say if I did a frontal section, then because this is moving in and out of the plane of section, it looks like that's a bit. this is maybe a separate structure and that's maybe a separate structure and this is disconnected. Whereas they are highly related. They are all the same structure. So keep in mind that we're looking at a two-dimensional representation of what typically are three-dimensional objects. So remember the basic terms: a longitudinal section, it's cut along its long axis; a cross section, perpendicular to its long axis; and an oblique is sort of an angle between a cross section and the longitudinal section. And often your slides, when you look at them in histology, will give you an indication as to what type of section has been made. And you can just see some examples here of longitudinal, cross, and oblique sections. Also, when we are preparing histology, sometimes we just do a smear, and a smear, the tissues just spread or rub across the slide. And an example would be blood. So this works very good for very liquid type of tissues, and of course the main one would be blood. A spread itself is some membranes and tissues have a lot of pliability to them, and they can actually just kind of spread out onto a slide. And areolar tissue, we're going to see, is a type of connective tissue, and this one certainly can be spread out on a slide. Now, the next video will pick up and start to work its way through the major groups of tissues. We'll start off with epithelial, and then we'll continue on with others.